Hello everyone, I'm Hadi Skarache. I'm a PhD candidate in chemical engineering at NGIT. I'm honored to present to you today another study from Cosmo entitled Effects of T-consciousness Fields on In Vitro Maturation of Mouse Oocytes, Mitochondrial Membrane Potential, and Embryo Development Following IVF. This research investigates the intersection of infertility treatments and consciousness fields, offering insights into novel approaches that could impact fertility treatments. Let's delve into the details of this study together. Infertility is a global concern, affecting 8 to 12% of couples worldwide. Treatments like IVF have become increasingly common. However, it's essential to recognize that IVF isn't suitable for all patients. Factors like high antral follicle count or polycystic ovary syndrome can complicate its effectiveness. Let's talk about the world of T-consciousness fields, pioneered by Muhammad Ali Taheri. These fields are subcategories of the cosmic consciousness network. Through scientific experiment, TCFs have shown incredible insights into consciousness as a non-physical entity. What's particularly intriguing is their potential to transmit information, influencing the behavior of treated samples. So what's our focus here? We are looking into the effects of these T-consciousness fields on the in vitro maturation of mouse oocytes. Additionally, we're examining how TCFs impact mitochondrial membrane potential and embryo development through IVF. With this brief introduction, let's jump into methods of a study. For method, we have the application of TCFs. To ensure the integrity of our research, we strictly adhere to the protocols established by the Cosmo Research Center, which you can find details on their website, cosmointel.com. In order to maintain objectivity, we implemented the double-blind method. This means that lab technicians were unaware of TCF's theory, and the individuals applying the treatment were unaware of the study's detail. In this research, TCFs were applied at the beginning of the experiment. This figure shows the main part of the TCF application protocol, where the information is transmitted through announcer and cosmic consciousness network to the object under study. Now let's break down the experimental setup for both IVM and IVF experiments. For the IVM, we divided the immature oocytes into 5 experimental groups. First group is the negative control. A second group is positive control, involved the addition of inducing granulosal cells to the culture medium. And the remaining 3 groups were our treated samples, inducing by 3 different types of T-consciousness fields. For IVF experiment, to ensure successful fertilization, the sperm suspension were pre-incubated for capacitation before introducing it to the collected oocytes. Following this, the combined sperm oocyte suspension underwent incubation for 4 to 6 hours. Fertilization rates were calculated by counting the number of 2-cell embryos 24 hours post-completion. Now, let's look at the results. In comparison to the negative control, the positive control experienced a reduction of 20% in the average number of immature oocytes. This reduction was even more pronounced in the TCF-treated groups, with around 33%, 20%, and 36% observed for TCF1, TCF2, and TCF3 respectively. Significant enhancement in the maturation in metaphase 2 stage were observed in the TCF1 and TCF3 groups, showing the average increase of around 40% compared to the negative control. And when considering the sum of the metaphase 2 and germovesical breakdown stages, a significant change were observed under the influence of all 3 types of TCFs as well. The mitochondrial membrane potential of metaphase 2 oocytes was estimated using JC1 aggregate fluorescent intensity. We observed a significant difference in the values of the positive control compared to the negative control, with an increase of approximately 82%. Likewise, for TCF-treated samples, this number is 61% for samples treated with TCF1 and 69% for TCF3. In vitro fertilization outcomes in TCF-treated samples and controls are shown in this table and bar charts. Results indicate that the application of TCFs led to a significant increase in the percentage of 2PN zygotes, showing an around 12% increase compared to the control group. Additionally, TCFs demonstrated a significant impact by reducing the number of unfertilized oocytes by 10% compared to the control group. This table shows the percentage of different grades of embryos in control and samples under TCF's treatment. The percentage of A and B grades significantly increased under the influence of TCFs, 2 times higher than the control group. Also, a reduction of about 45% in the BC grades, which is indicative of low quality embryos, were reported as well when exposed to TCFs. To recap, I'd like to highlight that the impact of TCFs on IVM and IVF is explored for the first time in this study. IVM results showed improved oocyte maturation with increased mitochondrial membrane potential, and IVF experiments demonstrated that TCF treatment boosts the likelihood of successful zygote formation and also enhances embryo quality, suggesting potential benefits of T consciousness field exposure. Further research is suggested to validate the efficacy of T consciousness fields. At the end, I want to express my special gratitude to Muhammad Ali Taheri, the founder of Cosmo-Intel and innovator of T consciousness fields, and to our esteemed co-authors for their invaluable contributions to this study. With that, we would like to invite laboratories and research institutions for collaboration, and thank you so much for your attention.